Drug discovery is a long and complex process. Each step, from identifying a biological target to developing a safe and effective therapy, carries significant risk. That is why improving the reliability of target validation is so critical. It helps reduce failures. Today, with the help of AI-driven data analytics, we can leverage real-world adverse event reports to reveal how drugs interact with biological targets. In today's demo, we will explore one key question: how can we effectively use adverse events data for target validation during drug discovery? In simple terms, adverse event data helps us navigate whether a drug target truly derives the biology we care about. Target validation is about proving that modulating a specific protein or enzyme will have the desired therapeutic effect. And by applying data analytics across drugs that hit the same target, we can detect consistent patterns in adverse events. Patterns that increase our confidence that pursuing this target will yield predictable outcomes. So, what is the objective of today's demo? It is to show how adverse event data can be turned into evidence for target validation. The process has 4 steps. First, we gather publicly available safety datasets. Then use Python to extract drug adverse event pairs. Next, we run similarity analysis to see how different drugs compare. And finally, interpret the biological meaning. That is, identifying consistent target-driven patterns, spotting outliers, and evaluating off-target effects. At the end of today's demo, we will have answers to the key questions listed below. By addressing these, we achieve the demo's objective, that is demonstrating how adverse event data can provide clear, data-driven evidence for target validation. Questions are basically, do drugs acting on the same target show a consistent adverse event signature? Which drugs deviate as outliers? And how much of the pattern is target-driven versus off-target? So the drugs that we are going to use for today's demo are sulfasaline, aspirin, salicylic acid, and mescaline. All of them have a common target, the CXO enzymes, SOCOX1 and COX2. And the data source that we are going to use is the FDA's Adverse Event Reporting Systems quarterly data extract. Now let us start the demo. Let me first acquire the raw data adverse event data. Here I'm opening the FDA website and navigating to quarterly data extract section. These files contain publicly available safety reports. I have downloaded one of them and let's take a look inside. As we can see, each file holds hundreds of ICSR R2 XML cases. These are individual adverse event reports. This is the source data we will be working on with for our demo. Now, let's move to the data extraction step. We will be working with the R2B, E2B R2 XML files, which contain structured data on patient information, drugs, reported adverse events. Our objective is extract drug adverse event pairs and count how often they co-occur. Using Python, we will be looping through multiple R2 cases and tabulate all such drug AE pairs. The focus of this demo is on 4 drugs: aspirin, sulfalazine, salicylic acid, and mescaline. The Excel file is created. As we see here, the drugs and the corresponding AE terms are clearly documented. We have aspirin, mescaline, salicylic acid, and finally sulfasaline. The Python code used here is quite complex and I had to use GPT-5 to generate it. Now that we have extracted adverse event data for drugs of our interest, the next step is similarity analysis. The idea here is if multiple drugs act on the same biological target, they should produce similar patterns of adverse events. By comparing these patterns across drugs, we can test whether the target really drives the observed biology. To do this objectively, we will use cosine similarity, Janssen-Shannon divergence, Spearman rank correlation. This steps provides us with quantitative framework to evaluate whether adverse event data supports the target hypothesis. The output is now being generated. All files are generated successfully. Let me open the similarity score. As we see here, this particular row item has a very high similarity. The one with salicylic acid, however, appears to have low similarity. Now that we have extracted adverse events data and performed similarity analysis across our 4 drugs, we move to the final and most insightful step, biological interpretation. At this stage, our goal is to translate similarity metrics into actionable biological insights. Using tools like ChatGPT, we can systematically derive AE signature consistency and assess confidence in target-mediated biology. I have the prompt ready here. By using ChatGPT, we can now rapidly interpret complex data sets and generate insights that would otherwise have taken much longer to uncover manually. This step will directly answer the key questions we set out to explore in this demo. Let's look at response 1. Yes, this is the summary table with its interpretation. Aspirin and mesalamine show a very strong AE signature. Mesalamine and sulfasalazine also show strong alignment. Whereas we see the salicylic acid show very weak similarity, and in fact it is negligible when it comes to salicylic acid mesalamine. The short summary is also provided here. A core AE signature cluster has emerged between aspirin, mesalamine, and sulfasalazine, whereas salicylic acid has consistently shown low similarity. Now we are going to get the answers to the questions that we had asked ourselves earlier in the demo. Do multiple drugs hitting the same target show consistent AE signature? Yes. Aspirin, mesalamine, and sulfasalazine do show consistent and high A profile similarity. The second question was, does this consistency across drugs strengthen our confidence that a target derives these effects? The answer again is yes. High cosine values between the 3 drugs here suggest a reproducible AE fingerprint target which is shared to this particular target. So which drug deviates from the common signature? Salicylic acid actually deviates. The next question is, can AE similarity metrics, that is the cosine, JSD, correlation, etc., help us quantify what is target related versus what is off target? So again, we have got answer to this question here, and the answer is yes. And finally, does the shared AE signature provide biological meaningful fingerprint that supports our validation? And the answer is that the clustering of aspirin and these other two drugs provides us a reproducible fingerprint. Here, I have also asked the GPT to provide us the strategic next steps. The first one, of course, is the cluster confirmation. So we will have to use high similarity score as the core signature reference here. The next would be the outlier analytic analysis. In this case, it would be to investigate salicylic acid further. Continue with our metrics and continue with our validation experiments, whether it actually aligns to what we have understood so far. So now, in summary, we started with raw adverse event data, extracted drug event pairs, ran similarity analysis, and we have finally derived the biological insights. Together, these steps demonstrate how real-world safety data, enhanced by AI-driven interpretation, can be transformed into meaningful evidence for target validation. Integrating this AI-enabled approach into early discovery pipelines can help reduce failures and prioritize promising